Hello, and thank you for joining us. My name is Dr. Kristin Hennessy-McDonald, and I'm a curriculum specialist with Mini PCR. I'm here with Mini PCR Bio to introduce our True Blue Bacterial Transformation Lab, our latest biotechnology lab. At Mini PCR, we are always striving to bring the best options to our customers. And with this lab, we found a way to introduce bacterial transformation into the classroom with minimal equipment and a robust protocol that can be completed in a class period. As a former life science teacher who taught everything from general biology all the way to AP and dual enrollment biology in my 12 years in the classroom, I tried all of the bacterial transformation kits out there. And I can say from experience that this is the easiest, fastest, and most teacher-friendly kit on the market. Before we dive into today's lab, I want to start with an introduction. If you're new to Mini PCR Bio, we make biotech equipment and a curriculum to make it easier to access the latest biotechnology tools. Our portable, durable, and most importantly, affordable equipment is used for a number of different applications. Our equipment is used by scientific researchers in the field, including scientists at the CDC, who use our equipment to monitor disease outbreaks. Amateur mycologists use our equipment to identify mushroom species that they foraged. We even have equipment on the International Space Station. Here is one of our namesake mini PCR thermal cyclers pictured in front of the iconic cupola window on the ISS. A major focus of our work is to bring biotechnology into the classroom. In addition to our biotech equipment, we offer innovative lab activities to give students real hands-on experience with biotechnology so that teachers can show students how our equipment can be used to solve real-world problems. At Mini PCR, we believe that biotechnology is a tool that can be used throughout the life science curriculum. Today, we're going to be talking about bacterial transformation, and we have a lot of tools that focus on a number of fundamental life science concepts. These include the genetic basis of disease, or conservation ecology. Now, bacterial transformation is an essential biotechnology tool. In essence, transformation is the uptake of foreign DNA by bacteria. Transformation occurs in nature, but scientists also introduce DNA into bacteria through transformation in laboratory experiments. Transformation is performed daily in many scientific labs. This transformative protocol enables scientists to study the functions of specific genes and the regulation of gene expression. And students are excited to perform labs, especially hands-on genetic engineering, where they can identify the phenotype of bacteria. But up until now, performing transformation in the classroom required expensive equipment and extensive teacher preparation. Teacher preparation for some transformation labs can take up to a week before the lab can take place in the classroom, and can take up to 2 to 3 class periods for students to complete. At Mini PCR, we never planned on creating a transformation lab unless we could create a lab that would significantly impact our customers. We wanted a lab that any teacher would feel comfortable using, regardless of whether they were picking up a pipette for the first time or they had extensive experience in transformation. The True Blue Lab accomplished all of those goals, including creating a lab that required minimal prep for teachers. We knew that one of the biggest hurdles to performing a transformation lab was the teacher prep time that's required. So we wanted to create a lab that did not require multiple days of teacher prep. Now, most commercial transformation kits require teachers to pour multiple types of plates in sequence, depending on the plate additives, then rehydrate the bacteria and allow them to grow for 8 to 24 hours. After that, teachers then had to streak those bacteria onto starter plates and allow those starter plates to grow for 24 to 48 hours. And only then could students start the protocol by selecting colonies from those starter plates. This can take up to a week before students can even start the protocol, is labor intensive, and requires significant teacher time and planning. It can also fail at a number of steps. Our lab slashes teacher prep time down to about 30 to 40 minutes and wait time down to about 1 to 2 hours before students can start the protocol. So you're not spending a week preparing for a lab in your classroom. We're able to decrease the teacher time due to the star of the TrueBlue lab, our fast bacterial cells. All that a teacher has to do to prepare these cells is rehydrate them with calcium chloride, and we're going to talk about why we use this to rehydrate the cells in a little while. Then incubate them in the fridge for 30 minutes to 2 hours. This gives you the time you need to prepare for your classroom schedule. And then students can start the transformation reaction. I'm going to show you this on my lab bench right here. So here you see my lab bench, just like you'd have in your classroom, all set up to rehydrate my fast bacterial cells. The first thing I'll do is put on gloves. You always want to make sure you wear your PPE when working with bacterial cells. Then what I'm going to do is I'm going to use my P1000 set at 650 microliters. And I'm going to take the calcium chloride that comes with the kit and pull up that 650 microliters. Then I'm going to take our fast bacterial cells, open them up, add that calcium chloride, put the top back on, and invert them a few times. The fast bacterial cells come lyophilized or freeze-dried. And so what we're doing right here is putting them into solution and introducing them to the calcium chloride so that they're ready to be transformed. And you can see here how the cells are already in solution. At this point, to prepare them for your students, you're going to take your P200 and you will have already labeled 20 Eppendorf tubes with that B for bacteria. You're going to open up those fast bacterial cells and take 30 microliters of those cells and put them in your pre-prepared Eppendorf tubes. Now I'm not going to sit here and make you watch me do all of these, but once I had completed putting my 30 microliters of fast bacterial cells into my Eppendorfs, I would just take all of these Eppendorfs and put them in the fridge. Again, they can be incubated for between 30 minutes to 2 hours before the students start their transformation. This gives you the flexibility to prepare the cells when you can for your students. So this easy 3-step process combines the rehydration and cell preparation steps from other labs, meaning that our lab does not require overnight incubations to prepare the cells for transformation and removes the need for starter plates. If you can pipette a set amount of liquid into a tube, you can set up this lab for your students in about 30 to 40 minutes. In addition to minimal teacher prep, this lab also has minimal equipment requirements. Another major hurdle to transformation in the classroom are the pieces of equipment required to have successful transformation. The TrueBlue lab does not require a heat block, or hot water and a hot plate, or a vortexer. The only required equipment for your students is a 2 to 20 microliter variable volume micropipette. The curriculum was also written to engage students at multiple levels, from students in a general biology classroom all the way to the AP level. On this screen, I'm going to go through a few pages of the lab guide so that you can see what I mean when I say this lab was written to engage students at multiple levels. This page shows how the information is broken down into easy-to-read units, with complex words defined to increase student understanding. Each text box on the page contains a set of specific and related information, and each page only has between 2 and 4 text boxes, making the information easy to digest. When writing this lab, we intentionally wanted to make it accessible to students without pages of dense, text-heavy background. In my classroom, students would vigorously protest when given long pages of background information when we were preparing for labs. And so when writing this lab, I really wanted to make sure that it was written with students in mind. Here you can see how the information is paired with easy to decipher diagrams. This example is the complex process of transformation, but you can see how it's explained in a diagram that breaks down each essential element. The text is paired with the diagram, making it easy for students to understand what is going on at each step. We also have assessment questions interspersed throughout the document. Here you see an example of one of our background stop and think questions. The information to answer this question can be found in the text and diagrams on this page, allowing students to search for the information and teachers a chance to check for understanding before moving on to the next concept. In many transformation labs, there's a combination of transformation and induction. This can cause confusion, blurring the distinction between the two processes. So the True Blue Transformation Lab focuses specifically on the process of bacterial transformation, allowing students to understand this important and complex process. This lab also has a robust and easy-to-read visual transformation output. Once students grab their plates from the incubator or from a warm, dry place in your lab, the students can easily see that they have successfully transformed the bacterial cells. At this point, I'm going to go through the background of the lab, just like you would in your classroom with your students. The slides that I'm about to present are available for free at minipcr.com, so you can use them in your classroom however you want with your students. I also want to point out that many of the illustrations in the slides come directly from the lab guide, so they give you a preview of the illustrations that I was just talking about. After we go through the background, I'm going to outline the experimental protocol that your students would perform in your class, and then we'll go over the expected results. Most bacteria have a single circular chromosome located in the nucleoid region of the cell. Bacteria can also have smaller circular pieces of DNA called plasmids. Now, both bacterial chromosomes and plasmids are copied by DNA replication machinery and passed down to daughter cells during cell division. Bacteria can pick up these small pieces of DNA from their environment through a process known as transformation. Now, transformation in nature is a rare occurrence. But once a plasmid has been transformed into a bacterial cell, the genes that it carries can be expressed by the cell that has been transformed. The maintenance of plasmids is expensive, so plasmids are only maintained by bacterial cells if they have a beneficial gene. One place that we see this in nature is with antibiotic resistance plasmids. Now, scientists use their knowledge of how plasmids in nature occurred and were able to engineer plasmids to introduce genes that they wanted to study into bacteria. Engineered plasmids always contain at least one gene related to what the scientist is studying. This gene of interest could be a gene for a protein the scientist wants the bacteria to produce. This is actually how most commercial insulin is made. It could also be a marker, like a protein that has a fluorescent tag, so that scientists can watch where it goes within a cell. There are a few methods that scientists use in the lab based on the bacteria and plasmid they're using. In our case, we're going to expose the cells to calcium chloride. This exposure has been shown to increase cellular uptake of plasmid DNA, leading to transformation of the bacterial cell. Now, just like in nature, transformation in the laboratory is rare, with few bacterial cells taking up the plasmid, even with the best of protocols. So while there are some bacterial cells that contain the plasmid, there are many more that do not. So we need a way to isolate and grow only successfully transformed bacteria. Typically, this selection is performed by including an additional gene, an antibiotic resistance gene, on the plasmid. So when the cell has been successfully transformed, it carries the plasmid, which carries the antibiotic resistance gene. This gene allows the bacterial cells to make a protein that inhibits the function of a specific antibiotic. So when treated with that antibiotic, the cells containing the plasmid can resist the effects due to the antibiotic resistance gene making that antibiotic resistance protein that again allows the cells to resist the effects of the antibiotic. So cells with the plasmid survive while those without the plasmid die. Following transformation, the bacteria need a surface on which to grow and food to give them energy. They tend to be spread on nutrient agar, which contains all of the nutrients that the bacteria need to grow. There are three possible outcomes following the introduction of bacteria to the agar. It is possible that there is no bacterial growth. In this case, you won't see any bacteria on the surface of the agar. And this tells you that somewhere in your protocol, all of the bacteria have died. The opposite possibility is that most of the plate is completely covered with bacterial growth. This is known as a lawn of bacteria because just like grass covers your lawn, the bacteria completely cover the surface of the plate. This is not really what you want when you transform because in that lawn, you can't identify the bacteria which have taken up the plasmid. The third possibility is that the cells grow in clusters. These clusters are known as colonies. This is because the cells in a colony are all derived from a single bacterial cell, which is divided a number of times. So to sum up what we've just gone over, it's possible to genetically engineer bacteria by introducing plasmids into cells. These plasmids contain, at minimum, your gene of interest and an antibiotic selection gene. Treating the cells with calcium chloride increases the likelihood that the bacterial cells will take up the plasmid. And because the plasmid confers antibiotic resistance, when grown in the presence of an antibiotic, it is possible to isolate only those bacterial cells which have successfully taken up the plasmid. In today's lab, students become genetic engineers and will engineer a bacteria to grow bright blue. We're able to accomplish this using the plasmid we engineered, the P-True plasmid. It contains the LAC-Z gene. That's our gene of interest, and I'll go over how it makes our cells turn blue in a minute. It also contains the AMP-R gene. This is our antibiotic resistance gene. As I said, our gene of interest is the LAC-Z gene. The LAC-Z gene encodes the enzyme beta-galactosidase. The beta-galactosidase enzyme has the ability to break down the substrate xgal. When xgal is broken down, it produces a blue pigment. Now, teacher to teacher, the clear student identification of blue colonies with no additional equipment makes this lab so easy to run in the classroom. As I said, as soon as the students look at their plates, they can see the difference between these bright blue colonies and all of their other cells. So, when bacteria are transformed with the plasmid, they have the LAC-Z gene. The LAC-Z gene allows the bacterial cells to make the beta-galactosidase enzyme. And when plated on nutrient agar containing that xgal substrate, the beta-gal is able to break down the xgal into a blue pigment, and the bacteria grow blue. There are only 2 transformations that students are going to be performing, and there's a reason for each treatment. So each group will get 2 vials or 2 Eppendorf tubes of those bacterial cells that we've aliquoted and put in the fridge. Our first reaction is a water-only negative control. This allows us to see that the bacteria are viable at the beginning of our experiment. Our other transformation reaction is the Petri plasmid. This is the plasmid that we're trying to transform the cells with. We're going to plate the bacteria on 2 types of plates. The first is plain agar. This allows us to see that the treatment with the Petri plasmid does not harm the bacteria. Our second treatment is our LB Carbex plates. We're using the antibiotic carbonicillin, which is an antibiotic that the AMP-R gene confers resistance to. So our LB Carbex plates, the CAR stands for carbonicillin, so we have our selection antibiotic, and the X stands for xgal, which is the substrate to beta-galactosidase. We're going to plate the 2 transformation reactions on both types of plates and monitor each for the type of bacterial growth. So this is the end of the background section. And again, the slides are available at minipcr.com for you to download for free. You can modify them to fit your classroom needs and go over the background with your students in your classroom. I'm now going to walk us through the experimental protocol so that you can see how easily your students can become genetic engineers. The first thing that happens once the students walk into the room is that they label the plates that they will use for bacterial growth. As I said before, prior to this, you will have prepared your cells using that calcium chloride and put them in the fridge and have tried to warm the plates to room temperature. So these students are going to take these plates and prepare them and label them for their cells. The remainder of the protocol offers some flexibility in incubation times. This lab was designed to be adaptable to your classroom needs, with protocol variations to fit most classroom schedules. This lab can be completed in as little as 40 minutes, with maximum transformation efficiency between 80 and 85 minutes. So you can choose the time points that fit best in your classroom. After the students have labeled the plates, they're going to get their bacterial cells and introduce the plasmid. Again, these bacterial cells have been incubated in calcium chloride. So they will then perform a 20-minute incubation on ice. This 20 minutes allows the cells time to take up the plasmid. After that 20-minute incubation, the cells are given a recovery media. There are two options at this point for your recovery step. The first is incubation at 37 degrees. I know, I said that this protocol didn't require a heat block. This step does not actually require a heat block because students can hold the tubes in their hands for this incubation. This is because 37°C is approximately human body temperature. Now, if you have a heat block or you have the time in your class to do a longer incubation, you can, but these cells can be ready to plate in as little as 10 minutes. Your other option is for the cells to recover at room temperature for up to 48 hours. So if this is a better timeline for your class period, you can have the students set up their reactions, have them add recovery media, and then let them come back the next day or even 2 days later to plate the cells. I want to point out, if you want to do an inquiry-based activity, this step is actually a great place to allow your students some of that flexibility. After your students have done the pre-lab, you can have them predict which time point and at what temperature they'll see maximum transformation efficiency. They can then be broken into groups based on their predictions because this lab has enough reagents for 10 lab groups. So once the students have done the lab, they can compare their results. The lab guide contains all of the equations necessary for students to determine transformation efficiency, so they can calculate and compare each method to determine which one was the best. Whenever you're done with your recovery step, the students will then spread the transformed cells on the warmed and prepared plates. At this point, plates can be placed in an incubator or kept in a warm, dry area of your classroom to allow the bacteria to grow. If they're placed in an incubator, students should be able to see results after about 24 hours. If you keep them in a warm, dark area of your classroom, successfully transformed cells can take between 48 and 72 hours to grow enough for observation. So you could perform this lab on a Friday, great fun Friday activity. Place the plates in a warm, dark area of your classroom. You want it dark because the X-gal is light sensitive. And then the students can come back on Monday morning and observe their plates. This is what students will see after that incubation on the plates. On the control plate with control cells, most cells survive. So you're going to see that bacterial lawn. This is because there's no selection, so they didn't die due to the antibiotic. But this treatment is important because it shows that the bacteria were alive when we started the transformation. On the control plates, the cells transformed with the P-true plasmid will also show that bacterial lawn. Again, there was no selection, so they all survived because there wasn't any antibiotic. This treatment is important to show that selection is required to identify successfully transformed cells. The students can't tell which of those cells have successfully taken up the P-true plasmid because there was no selection. On the plates containing the carbonicillin antibiotic, the control cells should not exhibit any growth. The control cells don't have the antibiotic resistance gene, so they are susceptible to the antibiotic on the plate. This treatment is important to show that the bacteria start out as susceptible to the carbonicillin antibiotic prior to that P-true transformation. On the plates that contain antibiotic, the cells transformed with the P-true plasmid will grow into easy to identify colonies. You can see how these cells are not growing in a lawn, but are growing in these small colonies. Because there's selection, only those bacteria which are transformed are able to survive due to the presence of the antibiotic resistance gene. This treatment is important to show that the bacteria have antibiotic resistance when transformed with the P-True plasmid. The plates also contain X-gal, which is the substrate for the beta-galactosidase enzyme. So on the X-gal containing plates, the cells transformed with the P-True plasmid should turn blue. Again, the X-gal is the substrate to the beta-galactosidase enzyme. So this allowed the cells to produce that bright blue pigment because the LAC-Z gene, which creates the beta-galactosidase enzyme in these bacterial cells, is present on the P-True plasmid. So with a protocol only requiring the use of a micropipette, students can become bioengineers and transform bacterial cells. This protocol allows students to easily visualize the transformed cells with no additional equipment, as it's very easy to see once they look at the plates that the bacteria have changed from white to brilliant blue. So let's tie this all together. We started with bacterial cells that were susceptible to the antibiotic carbonicillin and grew white. We transformed bacteria with a plasmid that contained the antibiotic resistance gene and the LAC-Z gene. The antibiotic resistance gene allowed the bacteria to grow in the presence of the antibiotic carbonicillin. And the LAC-Z gene allowed the bacteria to break down its substrate, the substrate of beta-galactosidase X-gal, and turn blue. As a reminder, our goal was to create a lab that required minimal prep for teachers, was able to be performed in a single class period, and had a robust transformation efficiency with an easy to visualize output so that students could very easily see the results of their work. With the True Blue Lab, we created a lab that fulfilled all of these goals, allowing you to introduce your students to the process of genetic engineering. The lab guides for this lab and all of our labs are available at minipcr.com. On the True Blue Lab store page, which is linked in the description below, you can find all the lab curriculum available to download for free. This includes the teacher's guide, the student's guide, and the editable classroom slides, some of which we went over today. It can all be found under the Curriculum Downloads tab. One of the newest things we've added is an e-worksheet. This is something that our teachers asked for. This is a Google Doc that you can download. You can edit the questions to fit your students' needs and assign it directly into your LMS. So rather than having to stand there and copy the whole student guide so that the students can answer the questions, you can just assign this in your LMS and the students can put their answers directly on that e-worksheet. Beyond this lab, at MiniPCR, we have a lot of additional free educational resources that we've developed to help you bring biotech to your class. On our education resources page, you'll find links to our large library of videos and webinars, including other live lab demos and seminars on hot topics in biology. We also have biotech tutorials, including worksheets on bacterial transformation, PCR, and gel electrophoresis that are great companions to our labs. In my classroom, I usually had students read these before I gave them the labs so that they could understand the process of what they were about to do. We also have a series called DNA Dots. This is a library of 2-page biotechnology explainers written at the high school level, which come with classroom-ready question sets. The page is accessible at the QR code here. This wraps up our in-depth look at our new True Blue Bacterial Transformation Lab. In the description below is a link to sign up for our newsletter, which allows you to be notified about new lab activities that come out from MiniPCR. I want to thank you all for joining me. Have a great day. Goodbye.